endothelial cells. So he's an endothelial cell expert. And, you know, the last couple of years, we've just seen such an amazing explosion of understanding of how LDL actually gets under the endothelium and does all the nasty things that many of the rest of us study. So welcome, Warren. And as per usual, everybody, we're going to wait until the end for questions. So just raise your hands and then we'll get to that at the end. So take it away, Warren.
Thanks. Thanks, Katie. Thanks you, Bruce, for the invitation to present. Can you hear me okay? Can everyone hear me? Yeah, okay. So as Katie mentioned, my lab works on endothelial permeability. And the literature in the field of endothelial permeability focuses on the generation of intercellular gaps in an endothelial monolayer, which leads to paracellular leakage and tissue edema, particularly in the context of inflammation, like lung injury and sepsis. But this focus on paracellular gaps sort of neglects the other potential route by which molecules can get out of the vascular lumen. And that is the notion that even in the absence of inflammation, molecules and even cells can cross not only between adjacent endothelial cells, but actually through individual endothelial cells by transcellular transport or transcytosis. And this is not a new possibility. It was recognized decades ago by pioneers, including George Palade, who did really elegant electron microscope work, where he observed innumerable vesicles inside the cytoplasm of endothelial cells. And in this case, this is a myocardial capillary. And he and others postulated that these vesicles actually served as a sort of shuttlecraft, carrying ligands from the vascular lumen across the cytoplasm of the cell and depositing them in the interstitium of the blood vessel. And he called this transcytosis. And he recognized that even though this was a possibility, that because of the nature of the studies they could do, that he couldn't really conclude anything about the relative importance of this potential route of permeability relative to, for example, the importance of paracellular leakage during inflammation. My own lab, which focuses on endothelial permeability, started being interested in this topic some time ago when I read reviews like this by Ira Tavis and colleagues that stated that subendothelial lipoprotein retention is the initiating process in atherosclerosis. And as a young faculty member, I thought, well, that's interesting. How does the lipoprotein get there into the subendothelium? Certainly, it was possible that LDL was crossing between adjacent endothelial cells, but it's known that the size of the molecule is far greater than the normal size of a competent endothelial cell-cell junction. And I wondered about the possibility instead that LDL might cross an intact endothelial monolayer by transcellular transport or transcytosis. And this possibility had been considered by Simonescu and colleagues, again, decades ago, where they injected human and rabbit LDL into animal blood vessels, did serial and very elegant electron microscope studies, and they found what looked like the LDL being internalized in vesicles, crossing the cytoplasm, and then apparently being deposited at the basal surface by transcytosis. But importantly, they never saw this LDL crossing intercellular junctions, suggesting that the LDL, if it crossed, was crossing by transcytosis, not by paracellular leakage or convection. Now, in fact, this notion is supported by pathological studies. It's been known for decades that early atherosclerotic lesions in humans are covered by an intact endothelial layer, particularly early on, suggesting that the junctions are competent, that the only way the LDL could cross is through individual cells and not between them. So this was something that my lab decided to take on. And at the time, it was very much a niche field. This is a graph of the number of articles in PubMed. This is a control showing that there are thousands of articles every year dealing with LDL itself. And on the bottom, we have the number of articles dealing with LDL transcytosis in the systemic circulation. And these are single digits, and the gaps indicate 0. So despite something that's very important potentially to early atherogenesis, there's very little work being done on LDL transcytosis in the systemic circulation. The reason for this is both philosophical and technical. By philosophical, I mean that it was unclear whether this was an important process, so it didn't attract a lot of investigators. But from a technical standpoint, this is a low frequency event that is difficult to study. Those intracellular vesicles that George Palade observed, those caveoli, are known to decrease in frequency when cells are passaged in culture, making it harder and harder to study with each passage. And of course, there are methodological limitations. EM is very elegant, but it's not very easy to do mechanistic studies using EM. And the standard method of studying endothelial permeability using Boyden chambers or transwells is very difficult and cumbersome. So for example, in the standard approach, you would seed a Boyden chamber with hopefully totally confluent endothelial cells covering this semi-permeable membrane, and thereby generating an upper chamber and a lower chamber. And you would measure the flux of your tracer, for example, LDL, across. But when you take measurements like this, of course, you have no idea which route the LDL is taking. And we had shown some time ago, as had others, that manipulation of the endothelial monolayer by pharmacological or molecular means often induces paracellular leakage, confounding your ability to measure and quantify transcytosis. And of course, if you're working with primary cells, it's traditionally been quite difficult to achieve a high transfection efficiency of such cells on the monolayer, which of course, again, confounds your ability to make mechanistic insights. Furthermore, the generation of knockout animals to study transcytosis is impaired by the lack of understanding of what regulates transcytosis. How do you know what to knock out? One of the pioneers in this field is Arnold von Eckartstein in Switzerland, and this is an elegant paper of his where he tried to get around this by doing a radio-labeled HDL transwell assay. Very elegant work, and what he does in these assays is he adds an excess of cold, unlabeled HDL to compete the labeled HDL, and then subtracts this, what he calls non-specific signal from the total signal to get a measurement of transcytosis. Of course, the problem with this approach, as you can see, is the non-specific signal is even greater than whatever you're measuring, which makes this a difficult technique, I would say, for mechanistic studies. So when we started in this field, the first thing we wanted to do was to show that an intact blood vessel performed LDL transcytosis. So we took a murine aorta, excised it, and perfused it ex vivo with LDL and with a biatenylated dextran molecule of a smaller molecular radius. And then after several hours of ex vivo perfusion, we fixed the vessel and probed for lipid using Nile red, and we could detect intimal lipid deposition, but no biatenylated dextran deposition, even though the dextran was smaller than the LDL. And this lack of dextran was not because it was somehow being repelled from the vessel intima, because when we opened up the junctions between endothelial cells using platelet activating factor, there was an abundant deposition of dextran, suggesting that the LDL that we had observed in the vessel intima had gotten there by transcytosis. So this was reassuring, but not very amenable to mechanistic type studies. So we then wanted to develop a single cell or a posse cell assay for transcytosis that would be useful for making mechanistic insights. So we verified that human coronary artery endothelial cells in culture maintain tight junctions and adherent junctions and some sort of polarity, whereby if you add LDL to the apical surface, over time, the LDL moves towards the basal lateral membrane. And we then developed a very simple assay based on total internal reflection fluorescence microscopy, or TIRF, which is based on the physical principle that when you shine a laser at a glass surface at a critical angle, almost all of it reflects, except for an evanescent wave of light that only penetrates the bottom 100 to 150 nanometers of a surface. So only this bottom of the cell monolayer becomes visible. And this reduces confounding from cytosolic or apical fluorescence. So the simple notion is you would add a fluorescent ligand like LDL apically, and then you'd be able to monitor its exocytosis at the basal membrane in real time. And the principle behind this assay has been known for decades. This is a paper from Nature where they used TIRF microscopy to follow exocytosis of a secretory granule in a live chromophim cell, showing brightening of the fluorescence and then its abrupt loss. So the principle is simple. You add fluorescent LDL apically to a confluent monolayer of endothelial cells. You don't see it until the vesicle enters the TIRF zone where the evanescent wave is illuminating. And then here, the fluorescent LDL containing vesicle can fuse with the basal membrane, releasing its fluorescent cargo, leading to a very rapid loss of fluorescence. Alternatively, the LDL can stay in the TIRF zone and undergo photobleaching, leading to a more gradual loss of fluorescence. or it may move back and forth in the TIRF zone, the endocytic traffic, so to speak, causing fluctuations. And you can quantify these events using a single particle tracking in MATLAB and incorporate a number of important controls. For example, to reduce confounding from endocytic traffic, you can include a parameter that only vesicles that dock and become stationary before losing their fluorescence are included. We only include vesicles that have a very rapid loss of fluorescence. So, for example, a change in the rate of fluorescence that is 3 standard deviations greater than the previous slope. And of course, we monitor images to make sure that for 5 additional time points, there's no reappearance of the vesicle of interest. And this script was originally written by Brian Height at Western Ontario and optimized for our microscope by my postdoc, Dr. Siavash Ghaffari. So using this principle, using this assay, this is what you see looking at the bottom of a human coronary endothelial cell. And this, as you can imagine, this is kind of exciting. You're actually monitoring transcytosis in real time in a primary cell. So using this approach, we showed that this assay is not affected by paracellular leak. So if we induce paracellular leak in the monolayer using platelet activating factor, transcytosis is not affected, that it's computable using an excess of unlabeled LDL, that it requires the large GTPase dynamin that's required for scission of these vesicles and internalization of these vesicles, because using the inhibitor DINGO almost completely ablates LDL transcytosis. And we were interested in identifying a receptor that mediates transcytosis in the systemic circulation. When we treat the coronary cells with recombinant PCSK9, we get almost complete degradation of LDLR, but there's no effect on the number of transcytosis events. So in the systemic circulation, at least, LDLR is not required. And instead, what we found was that there was co-localization of endogenous SRB1 with LDL, at least partially. We found that overexpression of SRB1 in the coronary cells induced LDL transcytosis, that even only partial knockdown of SRB1 significantly reduced LDL transcytosis. And as all of you know, the canonical ligand for SRB1 is HDL. And if you add an excess of unlabeled HDL, you also significantly attenuate LDL transcytosis. And then lastly, we took aortas from globally deficient SRB1 mice and perfused them ex vivo with biatenylated LDL for several hours, and then in a blinded manner quantified the accumulation of LDL in the aortic intima and showed a significant reduction of LDL accumulation in the aorta of these globally deficient animals. So we published this assay some years ago describing a role for SRB1 and LDL transcytosis. We also collaborated with Bill Sessa's group for their Nature Communications paper, helping them show that ALK1 is another receptor for LDL transcytosis. And many of you, of course, will have seen this tour de force from the Mineo and Shaw labs, where they knocked out SRB1 specifically in the endothelium using multiple murine models of atherosclerosis, showing that depletion of endothelial SRB1 significantly reduced atherosclerosis. And they also confirmed a role for SRB1 in LDL transcytosis using both TIRF microscopy and Transwell approaches. So with that as sort of wordy background, my lab has moved on to work on a number of projects, some of which I'm in a position to share with you today. So in addition to trying to identify some novel regulators of transcytosis, we've been focused on looking at the effect of traditional cardiac risk factors on LDL transcytosis. And the first one I'm going to talk about is the role of biological sex. So it's been known for many decades, probably hundreds of years, in fact, that premenopausal women have a significantly lower risk of heart disease than age-matched men. And this is not fully accounted for by differences in circulating cholesterol levels. The benefit of the premenopausal state has traditionally been attributed to estrogen. And in preclinical models, the benefit of estrogen also is not fully accounted for by changes in plasma cholesterol levels. And in fact, this study from JCI 30 years ago took postmenopausal monkeys and administered estrogen and progesterone replacement and showed reduced LDL accumulation in the coronary arteries, which was not due to differences in lipid levels, but they postulated was due to a direct effect at the arterial wall, but transcytosis was not examined. So Siavash Ghaffari in my lab took this and managed to get coronary artery endothelial cells from multiple donors, 3 males, 3 young females, and 1 postmenopausal donor, and measured basal rates of LDL transcytosis. And to our surprise, we found that on average, the rates of transcytosis were higher in the males than in the females. He then treated these coronary artery endothelial cells from male and female donors with physiological concentrations of estrogen, and found that in the males, there was a significant effect of estrogen to decrease LDL transcytosis, but no effect in the female cells. And this was not a non-specific effect on cell health or vesicular traffic, because in neither the male cells nor the female cells was there an effect of estrogen on albumin transcytosis. Now, along the same lines, the major protein constituent of caveoli is caveolin-1, and there was no effect of estrogen on either whole cell levels of caveolin-1 or on the subcellular distribution of caveolin-1, consistent with the albumin transcytosis data, because albumin transcytosis is performed by caveola. Instead, what Savage found was that estrogen significantly downregulates levels of the SRB1 receptor, as well as the LDL receptor, but had no effect on ALK1. And he found that this effect of estrogen and SRB1 was required for the diminution of LDL transcytosis. So while estrogen decreases LDL transcytosis in the male coronary artery endothelial cells, if SRB1 has been depleted, there is no effect. And he confirmed this by an alternative method where he overexpressed the SRB1 receptor in the male cells, and this overexpression significantly upregulated LDL internalization. And while estrogen downregulates LDL internalization in a dose-dependent manner in vector-transfected cells, overexpression of SRB1 attenuated this effect. We wondered whether this was a cell type or tissue type specific effect, so we looked at the effect of estrogen in liver-derived HepG2 cells from a male donor, and estrogen had no effect on SRB1 mRNA, but significantly upregulated LDLR mRNA and protein. And consistent with this, incubation with estrogen significantly stimulated LDL uptake in the male HepG2 cells, suggesting that maybe one beneficial effect of estrogen is to decrease LDL transcytosis by the endothelium and upregulate LDL uptake in the liver. To try to delineate the mechanism of this further, we looked at the estrogen receptors. And there are two classical nuclear receptors for estrogen, ER alpha and ER beta, and the anti or the protective effects of estrogen are traditionally ascribed to ER alpha. More recently, there's been a novel estrogen receptor, the G protein coupled estrogen receptor GPER, that has been described to have anti-atherogenic effects. And so we compared the expression profile of these estrogen receptors in the coronary artery endothelial cells and the liver cells, and found that GPER was significantly enriched in the endothelial cells. And when we blocked ER alpha and ER beta pharmacologically, there was no effect on attenuation of LDL transcytosis by estrogen. However, when we blocked GPER specifically, estrogen's attenuation of LDL transcytosis was prevented. More importantly, even if we only partially knocked down GPER by siRNA, the effect of estrogen on transcytosis was ablated. Now, GPER has been reported to signal via transactivation of the epidermal growth factor receptor. So if we inhibited EGFR using tirfostin or AG1478, this also blocked the effect of estrogen on LDL transcytosis and the effect of estrogen on SRB1 levels. So the model, as it stands now, is that estrogen decreases SRB1 expression through GPER and EGFR, and that this may contribute to the protective state of the premenopausal women against heart disease compared to age-matched men. And this area of how GPER relates to SRB1 expression is an ongoing area of research in my lab. A second story that's in progress in my lab is trying to look at the role of inflammation on LDL transcytosis. And this seeks to leverage the fact that the TIRF-based assay is not affected by the induction of paracellular leakage between adjacent endothelial cells. So it's sort of perfect for this type of project. And we have been initially interested in the role of a protein called high mobility group box protein 1, or HMGB1, that some of you may know. And it's actually the most abundant non-histone nuclear structural protein that there is. It's got 2 DNA binding boxes and an acidic tail. And despite this important structural role, it's known almost exclusively nowadays as an alarmin that is secreted by non-necrotic leukocytes, but also released by dying leukocytes. And when it's released, it can have cytokine and chemokine-like properties. It's known to be related to atherosclerosis, and in particular, late heart disease. So antibody inhibition of HMGB1 reduces disease in the ApoE knockout mouse model. And in humans, serum HMGB1 levels are correlated with the severity of disease. But we were interested in whether HMGB1 might have a role much earlier on in atherosclerosis, so whether it might regulate LDL transcytosis. And we were also interested in the specific role of endothelial HMGB1, because while in late disease, HMGB1 may be secreted or produced by endothelial cells, smooth muscle cells, and leukocytes, early on, its primary source would be the endothelium. So we wondered, what is the role of nuclear HMGB1 in the endothelium in LDL transcytosis and early atherosclerosis? So Siavash Ghaffari knocked down HMGB1 in primary human coronary artery endothelial cells. He did it in both cells of male and female origin, and found that LDL transcytosis was significantly attenuated, but there was no effect on albumin transcytosis. And the effect was obvious. This is the TIRF plane from cells depleted of HMGB1 compared to cells treated with a non-targeted control. And you can see a significant reduction in the number of LDL containing vesicles in the TIRF zone. To look at this by a complementary approach, he transiently transfected the coronary artery endothelial cells with GFP-tagged HMGB1, and this significantly induced LDL transcytosis. But at this point, we didn't know whether this effect was due to perhaps secretion and an effect of extracellular HMGB1, or whether we were still looking at nuclear or intracellular HMGB1. And to try to distinguish the two, we used glycyrrhizin, which is known to bind to HMGB1 and inhibit it. And so overexpression of HMGB1 in the coronary cells induced transcytosis, but inhibition of HMGB1 with glycyrrhizin had no effect. And along the same lines, we depleted one of the main receptors for HMGB1, RAGE, by siRNA. And this also had no effect on induction of LDL transcytosis, suggesting that overexpression was working through an intracellular pathway that we became interested in pursuing. So the TIRF zone images suggested that this was a decrease in uptake, probably. And indeed, when we knocked down HMGB1 by siRNA, we observed a significant reduction in SRB1 protein levels in the coronary cells, which was really intriguing. And at this time point, there was no difference in LDLR expression. And we confirmed this by qPCR, where we found that knockdown of HMGB1, of course, reduced SRB1 mRNA. It also reduced SREBP2 mRNA, but it had no effect on ALK1 or caveolin-1 or CD36. To try to figure out whether SRB1 and HMGB1 might be working in the same pathway, we did combinatorial knockdown. So while individual knockdown of HMGB1 or SRB1 significantly reduced LDL transcytosis, combined depletion had no additional effect, consistent perhaps with them working in the same pathway. I showed you in the previous slide that when we knocked down HMGB1, we also observed a significant reduction in SREBP2, like critical cholesterogenic transcription factor. And we started to wonder whether HMGB1 depletion was affecting SRB1 through an effect on SREBP2. And Thierry Huby had shown some time ago, in fact, that the SRB1 promoter contains SREBP binding sites. And in fact, when we depleted HMGB1 by siRNA, we found reduction in both the precursor and the mature form of SREBP2. We then did combinatorial knockdown of HMGB1 and SRBP2, and individually these reduced LDL transcytosis, whereas combined depletion had no additional effect. To try to implicate them being in the same pathway by an alternative approach, we did overexpression studies where we overexpressed HMGB1, induced transcytosis, but this benefit or this increase in transcytosis was dependent on SREBP2, because when SREBP2 is depleted, the effect was lost. We wondered, given this story, whether LDL was having an effect on HMGB1 translocation or abundance or mobility in the nucleus, whereby it might affect SREB2 function. And so we looked at the subcellular localization of HMGB1 in response to LDL. And so, not surprisingly, at baseline, most of the HMGB1 is found in the endothelial nucleus. But even with brief exposure to LDL, there was a significant increase in the nuclear to cytoplasmic ratio of HMGB1, quantified here. And this apparent translocation was at least partially dependent on the SRB1 receptor, because when we knocked down SRB1, you can see that there is a partial attenuation, suggesting that there's some sort of sensing mechanism that's causing the translocation. To verify this by an alternative approach, we did fluorescence recovery after photobleaching or FRAP assays in the coronary cells that had been transfected with GFP HMGB1. And this is an assay for mobility of the fluorescent protein. So we did this in cells that were exposed to LDL or not. And we found that there was a significant increase in HMGB1 mobility in the nucleus upon exposure to LDL, consistent with our immunofluorescence data. And going through the literature, this fit with the observation that others had made in the context of other transcription factors, where HMGB1 has been shown to enhance the activity of transcription factors like the steroid hormone receptor, like P53. And in fact, we found this paper that HMGB1 actually enhances SREBB2 function. So we wondered whether, in addition to enhancing function, nuclear HMGB1 might be regulating SREBB2 stability and maybe its half-life. So in this experiment, what Siavash did is looked at the half-life of SREBB2 mRNA in cells depleted of HMGB1 or not. And when HMGB1 was absent, the half-life was reduced by almost 50%, which was an interesting, although the mechanism of this remains to be determined. Now, to try to translate these in vitro findings to in vivo is a bit challenging because, of course, endothelial HMGB1 will have other functions besides regulating transcytosis. But we figured that the best way to do this would be to take endothelial deficient HMGB1 mice and then perfuse them, inject them acutely with fluorescent tagged LDL, and then after a very short period, look at LDL accumulation in the aorta, because we reasoned that this would reduce confounding from non-transcytosis effects, for example, effects on leukocyte recruitment and so on. And so we generated type 2 Cre-expressing HMGB1 flox-fluxed animals and their littermates, injected them with Alexa LDL, and 30 minutes later looked at LDL accumulation in the aorta. And although preliminary, these data show that there's a decrease in uptake of LDL in the inner curvature of the aorta of these mice. And this is not due to a difference in the endothelial tightness of these knockout animals compared to controls, because when we look at the accumulation of albumin using Evans blue, there's no difference. And of course, albumin is a much smaller molecule than LDL, so it's a more sensitive readout anyway for paracellular leakage. So this is consistent with our in vitro data suggesting decreased transcytosis. We went on to try to induce atherosclerosis in these animals by feeding them for 18 weeks using a Pagan diet, because as you know, these wild-type animals don't generally develop atherosclerotic lesions. And in fact, only half of them did, despite 18 weeks on this diet, only half of them developed neutral lipid accumulation. But of those that did, the endothelial deficient HMGB1 knockout animals had significant reduction. And this is despite no differences in total cholesterol, LDL, or triglycerides in the knockout and littermate controls. We then generated a double knockout animal on an LDLR deficient background. So mice deficient in HMGB1 in the endothelium, as well as LDLR globally, were fed a high-fat, high-cholesterol diet for 5 weeks. They had a significant reduction in neutral lipid accumulation, again, despite no difference in total cholesterol, and perhaps a trend towards increased triglycerides and increased LDL levels in the double knockout. And this work, of course, is still ongoing. So to close, I believe that how LDL crosses the endothelium has been relatively understudied, that this TIRF-based assay is quantitative and easy, and it's well suited for mechanistic studies as it's not confounded by paracellular leakage or low transfection efficiency. We now know that SRB1 is one of the receptors that mediates LDL transcytosis, and there's a huge push on to try to understand the regulation of this event. We've shown that estrogen at physiological concentrations inhibits transcytosis via GPER and SRB1, and this may contribute to the lower rates of atherogenesis in premenopausal women. In ongoing work, we've shown that HMGB1 translocates the nucleus very rapidly upon exposure to LDL, where it regulates SRB1 expression, likely via SRBB2, and that loss of endothelial HMGB1 decreases acute LDL uptake in the aorta, as well as neutral lipid accumulation in mice, with of course the caveat that we can't attribute all of this to decreased transcytosis, especially in advanced disease. And this is a visual abstract here. So to finish, I'd like to acknowledge the members of my lab that work on the LDL project, particularly Siavash, Erica, Franoshi, Michael, and previously Susan and Changson, my sources of funding. And these are the people who have done almost all of the work with regards to the ongoing HMGB1 story. And I'm looking for a postdoc, so if any of you want to come to this tower and see what the city looks like from up here, please send me an email. I'd be happy to take any questions.
Oh, that was amazing, Warren. Thank you. Oh, Mireille already has the first question. I was going to say, raise your hands. Mireille is ready. Okay, go ahead.
Okay, hi, Warren. How are you? Really great talk, really interesting research. So I guess I have quite a few questions. So I think it's fascinating as well, LDL transcytosis. And, you know, I wonder, so a few questions. First, SRB1, do we know if it's more expressed on the apical or the basolateral surfaces of the endothelial cells?
It's expressed on both surfaces. I don't know that there's a preferential location because it may be affected by internalization, but it is known to be expressed on both ends.
Okay, so do we think that maybe the SRB1 expression by modulating plasma cholesterol levels would maybe affect caveolae and in such a way indirectly affect the cycling of LDL through the cell? Is it possible that just by modulating plasma cholesterol through SRB1 expression could affect the number of caveolae present and that way, you know, affect the transcytosis process or uptake of LDL?
It's a good point. You know, most of the experiments that we've done, we control for whether this is an effect on the receptor or just an effect on caveoli by looking at caveolar distribution and levels. So the data that I presented is independent of caveolae. The question of whether, what you're asking is whether by manipulating caveolar levels that might affect SRB1?
Well, more like, is it maybe affecting plasma cholesterol concentrations that SRB1 could be affecting the recycling of the LDL? So maybe not directly through the binding and internalization. Like, has the binding been shown as well?
Thought you were getting at that. So, you know, in the Nature paper, they identified a number of mutations that affected LDL binding and uptake. And that was also shown in the structural description of LIMP2. And there are a number of papers, including mine, that have shown, you know, uptake that's SRB1 dependent. But the exact, and there's even a paper in the Journal of Lipid Research a couple of decades ago that tried to generate a KD for that binding. But where the binding occurs, I don't think is known. It's not ApoB. So I think I think, you know, the current state of the literature is that it is binding to LDL, but we don't know where.
Yeah, I think it's fascinating because you also mentioned LIMP2 and the HMGB1 work is really interesting. So HMGB1 and LIMP2, we have recently found to be present at the surface of lipid droplets. So we've been looking for regulators of lipophagy. And if we silence them in foam cells using siRNA, they both reduce cholesterol efflux. So knocking down HMGB1 and knocking down HMGB2 reduces cholesterol efflux, consistent with your data that, you know, it may. So I'm just wondering if there's an indirect effect of cholesterol efflux and cholesterol concentrations in the membranes that would affect the whole transcytosis process.
So, you know, I think that's a great question. And one of the things that I alluded to, but I didn't go into because it's not fully developed, is this notion of how does the cell sensing the LDL that we're giving it to translocate the HMGB1 to the nucleus? And, you know, the Schall group had published maybe a decade and a half ago, that SRB1 was a plasma cholesterol sensor through its transmembrane domain using that Q445 mutant. And so I think that's definitely potential for part of our story. But even in the absence of SRB1, we get some translocation. So we think that there are probably additional mechanisms by which the cell is sensing this. And so I think you and I should probably discuss this further.
Yeah, definitely. Thank you.
Okay, maybe we'll do Mabruka first, and then we'll do Ebru, okay? Go ahead, Mabruka.
Hi, it's Mabruka Al-Qaidi from LSU Health Shreveport. So I really enjoyed your talk. I have two questions actually. So SRB1 expression, do you think early on in atherosclerosis, it changes in expression of the receptors, or is it changes in the affinity of the receptors toward LDL rather than HDL?
No, I don't think there's a change in its affinity. I think, I mean, so this is speculation because it hasn't been shown by anyone yet, but I actually wonder whether there's competition between HDL and LDL for SRB1 binding. Then in the presence of high LDL concentrations, you probably get saturation of LDLR, and then you get SRB1 and potentially ALK1 mediated transcytosis. And then later on in the disease, the whole thing's confounded because, of course, leukocytes will come in and they'll be bearing SRB1 as well. So, it's a difficult model to study, but in the very earliest stages of the disease, I think I do think that endothelial transcytosis is playing a role.
Thank you.
Okay, Ebru, go ahead.
So Warren, I have a question about the ongoing HMGB1 project. So I remember that HMGB1 can induce ER stress. I'm not sure about endothelial cells, but definitely in liver cells. And ER stress also drives SRABP1 and SRB, expression changes. Yeah. So I wonder if you considered that into your model.
We have indirectly, in that we know that albumin transcytosis is unaffected. So this isn't a non-specific effect where we're just making the cell unhappy and there's less transcytosis. We know that when we measure vascular permeability or transendothelial electrical resistance, there's no difference. So it's not like the cell is just shrinking. I think that in the basal state, the vast majority of the HMGB1 is nuclear rather than cytoplasmic. So the effect that we're having is due to changes in nuclear HMGB1 rather than affecting the cytosolic HMGB1 pool. So I don't know if that answers your question, but I don't think it's a non-specific response.
So I wasn't thinking of the cells being stressed, but I was thinking that HMGB1 leads to SRABP2 cleavage. That would regulate your cholesterol levels in your cells. So it might have an effect because of its regulation on the cholesterol.
Right. So
you might want to look up those studies to see how relevant they are to you.
Yeah, I think that it helps address one of the reviewer's points. Thank you.
I'm glad.
Okay, if there are any questions, I'd like to thank Warren. That was really excellent and really stimulating. And on to our next speaker.
All right, so I'm going to introduce Ling Yang. Well, thank you, Warren. It was a great talk. And Ling Yang is at the University of Iowa. Prior to that, she was, can you guys see me? Okay, she was a postdoctoral fellow in the same lab that I was at Harvard School of Public Health. Ling works in organelle stress, particularly ER stress response. And she's going to talk about this response in the liver. So I'm going to hand it over to Ling. Are you here, Ling?
Yes, I'm here.
Okay, go ahead.
Thank you so much, April, for your kind introduction. And I cannot thank you enough for the organizer to organize such amazing seminar. I learned a lot from this seminar, and also it's my honor to present our work in this audience. Although I'm a liver, focused lab, and I hope I can get ideas and guidance and comments from all of you. And you can see, can you see my screen? Okay, okay. So So our lab is interested in studying the interaction between chronic inflammation signaling and the organelle function in the context of obesity. So the organelle stress response that we are interested in study. Sorry, is there something wrong? Can you say that? Oh, now it's okay. Where I'm interested in study is is the autophagy and the unfolded protein response. And study from my previous mentor's lab and also many other groups have shown that obesity resulting in defective hepatic autophagy, which is coexist with a decreased adapted unfolding protein response in the liver. So together, those impaired organelle stress response contribute to disturbed ER homeostasis and also obesity-associated metabolic dysfunction. The question remains, what is the physiological cause for this impaired organelle function or inability to initiate organelle stress response in the context of obesity? I think I have difficulty here. So the inducible nitric oxide synthesis and the immediate excess levels of nitric oxide production has been considered as a chronic inflammation marker that involved in pathogenesis of many human liver diseases, that including obesity associated mild steatosis and also progressing toward NASH or other liver disease such as cirrhosis. So as you know, the nitric oxide is a gas molecule that has important role for to modulate vascular dilation, and this gas also has important role to modulate protein function through protein modification. So one of these critical protein modification mediated by nitric oxide is called protein nitrosylation. That is usually referred as the NO group binding to the free sign of a cysteine residue of protein. So intracellular